CRISPR and cerebral palsy. Let's discuss CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It is a gene editing tool developed by Emmanuel Charpentier and Jennifer Doudna. It works by utilizing a DNA cutting enzyme called Cas9. A gRNA binds to a designated area in DNA, and the pre-designed gRNA guides Cas9 to the designated part of the genome. Cas9 then cuts the DNA. Through cell signaling, that part of the DNA is repaired, and sometimes that can lead to disabling a gene. Here I'll discuss a bit of history, symptoms, and treatments of cerebral palsy. It was first recognized and studied by an orthopedic surgeon named Dr. John Little in the 19th century. He theorized that CP was caused during a difficult labor and was a result of birth asphyxia. We know that CP is a neurological disorder caused by an abnormal development of the brain. Interestingly, in 1999, a study discovered that a gene was linked to spastic cerebral palsy within chromosome 2q24.25. In patients suffering from spastic CP, you'll see increased muscle tone in affected areas and difficulty in mobility. Symptoms in infants include stiffness, inability to roll over, no mobility of one arm and hand, inability to crawl on all fours, and overextension of neck and back, among other things. In terms of treatment, currently there is no cure, but improvement of gait is possible. Other treatments include individualized plans, which consist of therapies like occupational therapy, physical therapy, and even speech, since CP also affects speech, and usage of medical devices such as ankle-foot orthotics or a straightening board to straighten one's back and posture. There's also the use of Botox injections and neuromuscular electrical stimulators. A method for treatment that I chose to focus on was taken from a study conducted in 2017 that utilized BTXA and NMES to improve functional gait in spastic cerebral palsy patients. The use of BTXA reduced tone and acted as a junction block to pave the way for electrical stimulation of targeted muscles. BTXA, which stands for botulinum toxin type A, prevents release of acetylcholine from nerve terminals. In other words, it disrupts exocytosis, causing muscular paralysis. For clinical use, it only partially inhibits neuromuscular transmission. In the study, they found out that introducing NMES during this period stimulates a more controlled muscle contraction in spastic cerebral palsy. The results? The combination of BTXA and NMES resulted in improved gait. Neither BTXA nor NMES can stand alone. In order to improve overall gait cycle, both methods must be used. As of yet, cerebral palsy cannot be cured. Individualized treatment plans improve quality of life. Fortunately, relatively new developments have identified more specific causes of CP. A 2018 study predicted presence of spastic cerebral palsy using DNA methylation patterns. In a 1999 study, they discovered a gene linking to spastic CP in chromosome 2q24.25. So what about chromosome 2? Since the 1999 study, little investigation has been made regarding link between chromosome 2 and spastic cerebral palsy. What we know is that chromosome 2 is the second largest chromosome. It makes up approximately 8% of our entire DNA. The Q in 2q refers to the arm below the centromere, which is also the longer arm. Within Q2425, the gene was detected that led to spastic cerebral palsy. Identifying the location of a neurological disorder could make spastic CP a candidate for CRISPR-Cas9. So how does CRISPR come into play? I already discussed generally how CRISPR-Cas9 works. In terms of cerebral palsy, it can target the Q2425 segment of chromosome 2. The gRNA can bind to the targeted location and help guide the Cas9 enzyme to its location. Cas9 enzyme could remove the targeted DNA strand, and through cell signaling, DNA repair could ensue. Through this process, certain genes can actually be silenced, and this can be incredibly beneficial for those affected by spastic CP. Having found a gene for cerebral palsy can now qualify it as a candidate for CRISPR-Cas9, and hopefully it'll soon become a disorder of the past.